Welcome to Tomorrow Unveiled. For decades, the whole idea of radically extending human life, it felt like something out of, late-night infomercials, maybe some science fiction.
Or a playground for Silicon Valley biohackers. Right.
Exactly. But based on this, this massive stack of source material that our dedicated research team has put together, we are officially in a brand new era for geroscience.
It's really happening.
It is. So we're taking this comprehensive body of knowledge, all drawn from major recent breakthroughs, clinical, academic, you name it, and we're synthesizing it into what we're calling the Immortality Report.
And I think the most important shift, the one that really stands out in all these sources, is it's conceptual.
How so?
Longevity science is moving way beyond just slowing decline. That whole anti-aging mantra, it was often pretty superficial.
Right. It lacked clear goals, robust endpoints.
Precisely. We're moving to something much more specific, functional life extension. And this isn't just about adding more years to your life.
It's about quality.
It's about validating and actually demonstrating the restoration of complex biological function in tissues that we used to think were, permanently aged.
So bringing things back online, not just keeping them from falling apart.
That's it. It is a fundamental shift in focus from the sheer quantity of life to the verifiable quality of life. Yeah. What the entire research community now calls maximizing health span.
That framing is so crucial because if we're actually talking about restoration, well, then we need completely new tools, new therapies, a new infrastructure.
A whole new playbook.
So our mission here on Tomorrow Unveiled is to unpack all that source material across those three major areas. First, we're going to explore the cutting-edge science of just measuring aging, these revolutionary new tools that can tell you precisely how fast your biology is actually moving.
And the fun stuff.
Then the really fun stuff. Second, we dive into the actual breakthroughs in tissue and metabolic rejuvenation, the mechanisms that are now, actively turning back the biological clock at a cellular level.
And finally, we have to talk about the infrastructure.
Yes. Third, we'll cover the technological and economic infrastructure that's making this future possible, focusing on that exponential acceleration from AI and, some of the surprising socioeconomic implications of all of us living healthier, longer lives. Let's start with this measurement revolution because, as the old saying goes, you just can't manage what you don't measure.
And the field has matured, exponentially. It's a world away from the general wellness guessing games of the past.
Right. It's now highly evidence-based, all guided by these incredibly precise predictive biomarkers.
What has to be? This maturation is happening because we're facing a genuine societal crisis, which is the burden of chronic illness.
That's the driver.
That is the driver. Chronic conditions are what cause the loss of function, which is exactly the thing we're now trying to reverse. So the market, the clinical demand, it's all laser focused on finding earlier, more precise predictive markers than the one we've always used.
Chronological age.
Chronological age, which is, the most common but probably the least useful marker we have. Knowing you're 55 years old tells a doctor almost nothing about your real cardiovascular risk or your cognitive resilience.
Not compared to what we can measure now.
Not compared to what's in your cellular profile, no.
Okay, so if chronological age is out, what's in? What's the evolution here and the tools we're using to replace it? What's the real difference between that first generation of biomarkers and these new, powerful clocks?
The evolution, it really lies in a shift from measuring a static state to measuring a dynamic rate.
Okay, break that down.
First-generation epigenetic clocks, which are based on DNA methylation patterns.
These are the chemical tags in your DNA that turn genes on or off?
Exactly. Those early clocks, they just measured biological age. They would compare your methylation profile to a big reference population and give you one number.
You are biologically 47, something like that?
Right. But that number, it was often just too broad to be truly actionable.
That makes sense. It tells you where you are right now, but it doesn't tell you where you're going.
That's it, perfectly. The newer generation of clocks and the compiled source material emphasizes these again and again. They track the pace of aging, and the difference is profound.
So it's not just a snapshot.
No. Think of it this way. Comparing your biological age to your chronological age, that's like measuring velocity. But tracking the pace of aging is measuring acceleration or deceleration.
Oh, I see. So am I aging 1 year for every calendar year, or am I aging, say, 1.2 years for every calendar year?
Precisely. Are you speeding up or are you slowing down?
That concept, acceleration versus velocity, it makes the clinical significance immediately obvious. Can you give us an example of one of these pace clocks and why they're so much better at prediction?
The key examples that are consistently cited in the research are Grimmage and especially Dundon pace. These clocks aren't just looking at random methylation sites associated with age.
They're more targeted.
Much more. They are specifically trained to look at sites that are linked to key health outcomes and mortality risk factors. Things like inflammation, metabolism, even smoking status.
And the data backs this up.
The data is compelling. A faster epigenetic aging rate, as identified by Dundon pace, it predicts a significantly higher risk of all-cause mortality of heart disease, physical impairment, and cognitive decline. It just far outperforms any of the earlier models.
So it's like a real-time assessment of your biological depreciation rate.
That's a great way to put it.
And crucially, the sources all stress that aging isn't this like unified experience happening all over your body at once. You mentioned this concept of organ-specific aging.
And that's a critical insight. It's what guides all modern intervention strategies. Aging is profoundly heterogeneous.
Meaning it's different everywhere.
Wildly different. Our research team noted that different tissues age at completely different speeds. For example, your immune system might be biologically exhausted, a state we call immunosenescence, while at the same time your heart or your liver is still functionally robust.
And this is what makes precision medicine so necessary.
It's the whole ballgame. The true value of these sophisticated organ-specific pace tests is using the insights they give you. For instance, if your liver clock is ticking too fast, you can fine-tune highly targeted factors.
Like maybe cutting back on alcohol.
Or specific nutritional supplements, stress management, even training volume. You target that specific pace rather than just adopting some, generic anti-aging protocol that might not even affect the problem area.
Okay, so moving from these predictive pace clocks to functional assessment, let's zoom in on the newest player in this space. One that directly tackles the challenge of measuring function itself, not just molecular risk. The intrinsic capacity, or IC clock.
The IC clock is a major step forward. I mean, a really major step. It moves the whole discussion beyond those purely molecular markers to integrate what truly matters to a person.
Sustain human function, quality of life.
Exactly. Now, like the pace clocks, the IC clock is also derived from analyzing DNA methylation patterns in your blood or your saliva.
But the algorithm is different.
Very different. It's trained not just against age or mortality, but against this construct called intrinsic capacity.
And how is that construct intrinsic capacity defined? What is it actually measuring?
It's the sum of 6 key domains of function. These are the things that collectively define healthy aging and resilience.
Okay, what are they?
Mobility, cognition, mental health, vision, hearing, and overall nutrition and vitality. The methodology is really sophisticated. The algorithm identifies specific methylation sites whose expression patterns correlate very robustly with clinical assessments in each one of those 6 domains.
That feels vastly more comprehensive. I mean, it's more real-world applicable than just a generic biological age number. But this brings up a pretty fundamental question. How does a chemical mark on our DNA actually correlate with something like my hearing ability or my physical mobility?
And that is the core technical beauty of these advanced models. It's not a direct, cause and effect measure of, say, hearing loss. It's not that simple.
So what is it capturing?
Instead, IC captures the integrity of the system that supports all those functions. Think of methylation as reflecting the cumulative wear and tear on the systems responsible for repair, for inflammation control, for cellular maintenance.
So it's a proxy for overall system health.
It is. The methylation sites, the IC clock maps, they often sit near genes involved in systemic inflammation, in cell signaling, in mitochondrial function, the big picture stuff.
So if my overall systemic maintenance is failing, that failure will be reflected in a predictable methylation signature, which in turn makes me more susceptible to decline across all 6 of those capacity domains at the same time.
Precisely. A lower IC score means you have a reduction in your functional reserve and your resilience. And our research team noted corroborated findings from major longitudinal cohorts, like the Framingham Heart Study.
The famous one.
Very. And in that study, the IC clock outperformed all first and second generation aging clocks in predicting overall mortality risk. It's capturing the essential biology of resilience.
So if we look at the benefits that come with a high IC clock score, what are they telling us about that link between the methylation patterns and our actual function?
They correlate very strongly with clear, measurable systemic benefits. People with higher IC scores, they show better immune performance. They have significantly reduced systemic inflammation markers like CRP and IL-6.
And I'm guessing lifestyle choices are a big part of that.
Predictably connected to healthier lifestyle choices across the board.
Oh.
So the decline it measures, that loss of functional capacity, that's the reason why, as the sources confirm, Americans spend an average of 10 to 12 years of their lives in poor health. Measuring IC is literally measuring the gap between lifespan and health span.
So if this tool really does achieve the scalability that the research suggests, how does it fundamentally change how a clinician approaches wellness and aging?
It changes everything. I mean, everything, because it provides an objective, affordable, and sensitive endpoint for interventions.
An endpoint.
Right. Our research team suggests this tool is key for clinicians, for researchers, for policymakers. For a clinician, it can identify people whose biological pace and capacity are declining years, maybe even decades, before a major chronic disease event. It allows for preemptive intervention.
And for researchers.
It's a crucial and validated endpoint for longevity trials. If a new drug or a lifestyle change truly restores functional capacity, the IC clock should move. You should see the needle go back.
That makes perfect sense. We could finally stop relying solely on subjective patient reports of feeling better.
Exactly. The goal here, as it's articulated in all the collected sources, is to create a scalable, affordable assessment tool. Maybe even through something as simple as a dried blood spot test.
Which would dramatically expand access.
It would expand who can get this functional assessment, especially in underserved populations where chronic disease incidence is the highest. The ability to measure functional capacity cheaply and reliably, that's the foundation upon which this whole field of functional life extension has to be built.
Okay, so segment 1 gave us these incredibly sophisticated measuring tools. The ability to identify which tissues are declining and exactly how fast.
The what and the how fast.
Right. So segment 2 is where we dive into the functional restoration itself. The actual molecular and cellular mechanisms that are proving they can turn that biological clock back.
The how to fix it part.
The how to fix it part. Let's start with what has sort of become the poster child of longevity medicine. Cellular cleanup. Specifically dealing with those persistent harmful cells known as senescent cells.
Or zombie cells.
Zombie cells.
The approach here is highly strategic. Instead of just, treating the symptoms of aging, longevity medicine is now targeting a core root cause of decline, which is the accumulation of these senescent cells.
And just as a quick refresher, what are they?
These are cells that maybe they've reached the end of their replicative life, or they've suffered serious damage so they stop dividing. And that's a critical defense against cancer, which is good.
But there's a downside.
A big one. They are profoundly dysfunctional because they refuse to die through the normal process of apoptosis, or programmed cell death.
And instead of dying, they basically turn into these little inflammatory factories.
Precisely. They just linger, and they release this toxic mixture of pro-inflammatory signals, growth factors, and destructive enzymes. We call it the senescence-associated secretory phenotype, or SASP.
And that SAP, it actively accelerates aging in the healthy cells nearby.
It does, and it stalls all meaningful tissue repair throughout the body.
So the strategy to deal with them involves something called Senolytics. What's the state of play in Senolytic therapy development?
We've definitely moved beyond the early enthusiasm for just general-purpose compounds. I mean, while the initial human data on readily available things like Physitine and quercetin suggested they could work.
Things you can get as supplements.
Right. And they suggested they could clear senescent cells, maybe even improve mobility and epigenetic age markers. But the focus now is all on precision senolytics.
What does that precision look like in practice? What does that mean?
The goal is to develop compounds that are designed to remove only the specific harmful senescent cells, the truly inflammatory ones, while leaving alone any that might be, transiently present and actually helping with wound healing or tissue repair.
So not a sledgehammer, but a scalpel.
Exactly. And the research team highlighted a shift toward using specific high unmet need conditions as proving grounds for this mechanism. A fantastic example is the Phase 12 trial using the senolytic combination desitinib and quercetin, or D plus Q for secondary progressive multiple sclerosis.
MS? That seems like a big conceptual leap. Why MS?
The rationale here, it links cellular aging directly to the disease pathology. In MS, especially the progressive form, the brain's ability to repair the damaged myelin sheaths around neurons just stalls out. It stops working.
And the hypothesis is that senescent cells are to blame.
Researchers hypothesize that this failure is largely due to a chronic inflammatory and inhibitory environment that is maintained by senescent glial cells, the supporting cells in the central nervous system.
So these zombie glial cells are just perpetually pumping out that toxic SASP.
Correct. So if the D plus Q combination can selectively clear out those specific senescent glial cells, the inflammation should drop, and the brain's own native repair mechanisms can finally switch back on.
It's like clearing the rubble so the construction crew can get back to work.
That's a perfect analogy.
Yeah.
This trial is a pivotal test of the core senescence hypothesis in a very complex central nervous system disease. If removing a specific population of these cells allows the body to restart its own repair process, it validates the whole approach as true functional restoration, not just slowing decline.
That's fascinating. Okay, so that addresses the cellular cleanup side. Now let's pivot to metabolism and immunity, which the sources suggest are really the next big frontier for systemic reversal. First up, the disruptive trend of GLP-1 drugs.
Ah, yes.
They started as weight loss therapies, but the sources frame them as something much, much deeper, a full metabolic reset.
They absolutely are. And the high percentage of physicians reporting that they recommend or use GLP-1 receptor agonists, it just confirms they are transforming medical practice. This goes so far beyond just reducing body weight.
It's not about the number on the scale.
Not at all. These drugs are triggering a genuine metabolic recalibration, and it happens regardless of the patient's initial BMI.
So can we break down the physiological effects that make them a reset? What's actually happening?
Well, the clinical data confirms robust multi-pathway systemic effects.
Hmm.
GLP-1s substantially reduce chronic systemic inflammation. They normalize glucose and insulin sensitivity by acting on the pancreas and the liver.
Okay, those are big.
And crucially, they improve multiple cardiovascular risk markers, often significantly, even before major weight loss is achieved. This suggests they are optimizing the internal signaling networks that so often become dysfunctional with age.
I can see why physicians are now exploring these short-term reset protocols. It sounds like just foundational medicine.
It is. The idea is that maybe short-term, medically supervised use could be enough to break that vicious cycle of inflammation and insulin resistance that drives so much age-related disease.
So you create a healthy physiological foundation.
Right. A foundation which can then be maintained through lifestyle adjustments. It's about using a powerful pharmaceutical tool for a temporary systemic optimization, rather than creating a long-term dependency, though, of course, the research into the best protocols is still evolving.
Okay, now onto the immune system, where the most dramatic reversal highlighted in the report involves aged hematopoietic stem cells, or HSCs. This feels like the most powerful example of pure functional restoration we've seen.
It really is. I mean, HSCs are the master stem cells. They generate all of our blood and immune cells. And immunosenescence, the collapse of the adaptive immune system in old age, that's a primary driver of late-life mortality from infectious disease.
And reduced vaccine efficacy.
And reduced vaccine efficacy. As we age, these HSCs become exhausted. They don't die, but they lose their capacity to produce naive T cells, which are absolutely essential for recognizing new pathogens.
And instead, they start producing the wrong things.
They shift production toward inflammatory myeloid cells.
So the cell factory is still running, but it's churning out the wrong product. And that product is actually contributing to the problem of inflammation. What drives that fundamental shift?
The research team compiled data pointing to a dysregulation of branched-chain amino acid, or BCAA metabolism, as the culprit.
Okay.
Specifically, aged HSCs were found to be metabolically reprogrammed into a pro-inflammatory hyperproliferative state. They were essentially consuming and processing BCAAs, leucine, isoleucine, and valine at a different rate.
And that sent the wrong signal.
It did. It signaled them to prioritize that myeloid output over the critical naive T-cell differentiation we need.
Wow, that is a very precise metabolic flaw. So how did researchers manage to switch it back?
This is the breakthrough. They identified a simple, specific metabolite. It's called alpha-ketosacchaparate, which is a breakdown product of leucine.
And what do they do with it?
By treating the exhausted stem cells from aged mice, whose biology is functionally comparable to, say, an 80-year-old human, with this one metabolite, they forced the metabolic switch back to its youthful setting.
And the functional outcome, I mean, did the cells just look healthier under a microscope, or was there a real change?
Far from it. Functional capacity was fully restored. The treatment re-enabled robust production of naive T lymphocytes, effectively rebooting the adaptive immune system's ability to respond to new threats.
The implications of that are huge.
They're massive. The decline of this adaptive system is why a simple infection can often be lethal to the elderly. A functional reboot of T-cell production using this highly targeted metabolic intervention It offers a potential universal prophylactic against late-life infectious mortality.
And it could massively boost the effectiveness of existing vaccines in older population.
It could. It's a game changer.
That is a phenomenal finding. Let's move to another aspect of metabolic regulation, something that has historically been dangerous. But the sources suggest a safe new path is emerging: mild mitochondrial uncouplers.
Right, and this requires us to acknowledge the dark history of compounds like DNP or 2,4-dinitrophenol.
This was an old weight loss drug from the 30s.
It was. DNP was a mitochondrial uncoupler. So your mitochondria normally convert fuel energy into ATP, which is the cell's power currency. DNP would uncouple this process. It would force the cells to release that energy as heat instead of power.
It essentially turns the body into a furnace.
Exactly. And the safety margin was terrifyingly narrow. I mean, the difference between the dose that made you lose weight and the dose that caused uncontrollable lethal hyperthermia was minuscule.
Hence its ban.
Hence its ban. However, the new class of experimental compounds are mild uncouplers, and they're designed with a focus on non-toxicity and a very wide therapeutic window.
So if DNP was an explosion, these new compounds are more like a controlled leak, right?
That is the perfect analogy. The research team used the image of a hydroelectric dam. Normally, all the water, the energy, it flows through the turbines to generate power, or ATP.
Okay.
These mild uncouplers create a small, controlled, and safe leak in that dam. And this controlled leak forces the cell to slightly increase its overall energy expenditure, burning more fat and sugar to compensate for the lost energy, releasing just a manageable amount of heat.
So the immediate benefit is obviously treating obesity by safely increasing the basal metabolic rate, but what's the longevity application here?
Well, by gently nudging the cells toward this slight inefficiency, the compounds also appear to lower the membrane potential of the mitochondria, and that, in turn, reduces the production of reactive oxygen species or oxidative stress within the cells.
Which is a key driver of aging.
A key driver of cellular damage and aging-related processes, absolutely. Therefore, these mild uncouplers could support healthier long-term metabolism and potentially slow overall cellular aging. It's offering a safer, scientifically rigorous path to a true metabolic hack that people have been seeking for decades.
Let's conclude this segment on rejuvenation by looking at 3 specific complex tissues: cartilage, the heart, and the brain. Let's start with cartilage regeneration, which is so central to maintaining mobility and health span.
Articular cartilage, the tissue that keeps our joints moving smoothly, it has very limited regenerative capacity. That's what makes osteoarthritis such a chronic, debilitating, and difficult to treat age-related condition.
So what's the new insight here?
The research team focused on a specific enzyme that accumulates with age, 15-PGDH.
And what does 15-PGH do to cartilage?
Well, it increases with age, and crucially, it degrades key signaling molecules like prostaglandin E2 or PGE2, which normally promotes cartilage health and repair. So by inhibiting 15-PGDH with a small molecule inhibitor, researchers saw just dramatic regeneration of articular cartilage in aged mice.
Not dramatic.
It restored the tissue to near youthful levels of health.
And the critical finding here, which really challenges a lot of past assumptions about tissue repair, is that stem cells were not the mechanism of action.
Correct. And this is a vital point for treatment development. The effect was entirely due to the existing cartilage cells, the chondrocytes, shifting their phenotype.
Their behavior changed.
Their behavior changed. Aged chondrocytes often shift toward harmful subtypes that drive ossification and fibrosis, essentially turning the cartilage into bone or scar tissue. By inhibiting 15-PGDH, the researchers forced the chondrocytes to revert back to the healthy, repair-active subtype that maintains the extracellular matrix.
So it's a functional restoration of the aged tissue from within. That holds immense promise for treating the millions of people affected by osteoarthritis.
It really does.
Okay, moving to the heart, another organ very susceptible to systemic inflammaging. The research highlighted the role of a protein called heaven.
Right. Heaven is a protein whose plasma concentration increases significantly with age, particularly after age 60, and it's strongly correlated with a measurable decline in cardiac pumping ability.
And we know the mechanism.
The mechanism found in older male mice is pretty specific. Heaven promotes cellular senescence and inflammation within the heart tissue itself. It does this largely by upregulating the production of a ligand called CCL5, which signals immune cells to adopt a harmful pro-inflammatory state.
And that contributes to fibrosis and the thickening of the heart walls.
Exactly. So heaven acts as a molecular aging signal, driving a very specific, localized inflammatory reaction in the heart.
So how did the researchers intervene?
They used gene therapy, specifically an adeno-associated virus, or AAV, to selectively knock down the expression of HEBN in aged mice.
And the
The
results?
results were remarkable. Age-related damage was reversed, senescent cell biomarkers decreased, fibrosis and hypertrophy, that thickening of the heart wall were diminished, and the heart's pumping ability improved substantially.
So it demonstrates that reversing the effects of just a single age-related inflammatory signal can partially restore age-damaged cardiac function.
That's right.
Finally, let's address neuroprotection, focusing on Alzheimer's disease and the use of the cytokine GM-CSF, or sargramostim.
GM-CSF is a long-approved drug. It's used to stimulate the production of white blood cells.
Right.
It's now being investigated in clinical trials for mild to moderate Alzheimer's disease. The research team emphasized a key study where patients were treated with GM-CSF while researchers monitored a very specific biomarker. It's called UCHL1.
And UCHL1 is a marker for what exactly?
It's a specific and highly sensitive biomarker for neuronal cell death that circulates in the blood plasma. UCHL1 is an enzyme that's highly expressed in neurons. When neurons are damaged or die, this enzyme gets released into the cerebrospinal fluid and eventually into the bloodstream.
Making it a reliable proxy for the rate of neuronal attrition.
A very reliable proxy. And the trial revealed a staggering result. Treatment with GM-CSF significantly reduced the plasma concentration of UCHL1 by about 40%.
40%, that's a massive reduction. What does that imply about the drug's mechanism?
Well, the senior author of the study pointed out that this reduction brought the neuronal death marker levels in these Alzheimer's patients down to levels typically seen in early life. It strongly suggests that GM-CSF isn't just masking symptoms. It's reducing the underlying neuronal cell death. The likely mechanism is that it stimulates immune cells, particularly microglia in the brain, to become more efficient at clearing out damaged apoptotic and senescent neurons and plaques.
So it's halting the acceleration of age-related cognitive decline and allowing the remaining healthy neurons to function more effectively.
That's the theory. It confirms that these measurable blood biomarkers are incredibly sensitive to therapeutic interventions that target these fundamental aging processes.
The accumulation of evidence here in this segment, from the HSC rebooting to the HEBN knockdown, it really just underscores that functional restoration is the new norm for geroscience. It's not science fiction anymore. Welcome back to Tomorrow Unveiled. We've covered the measurements and the mechanisms of biological restoration. Now let's explore the infrastructure and the societal context that is making all of this progress possible.
Right.
We have to start with the single greatest accelerant in R&D today, artificial intelligence. Specifically, a platform the research team highlighted called Drug-CLIP.
Drug-CLIP represents a fundamental paradigm shift in drug discovery. We're moving from linear, costly screening to exponential digital matching.
Okay, what was the old way?
Traditionally, finding a drug involved conventional docking methods. You're simulating the molecular interaction between a drug candidate and a target protein in a specific binding pocket. That process is slow, computationally intensive, and highly constrained.
Like trying millions of unique physical keys and millions of unique physical locks, one by one.
That's the perfect analogy.
Yeah.
And Drug-CLIP completely bypasses that slow physical simulation. It uses deep contrastive learning to convert both the drug molecules and the protein binding pockets into high-dimensional digital vectors.
A digital signature.
Think of it as a digital signature or a mathematical fingerprint. Instead of simulating the physical chemistry, the AI translates the complex biological structures into math, and that allows it to compare the fit instantly through a high-efficiency semantic search in that vector space.
So instead of trying the physical key, it just compares the digital signature of the key to the digital signature of the lock to predict the fit with extreme accuracy.
Precisely. And the breakthrough isn't just the theoretical speed, it's the achieved scale. The compiled data noted that this platform performed 10 trillion molecule protein evaluations in a single day.
10 trillion.
On just one computing node. This compresses years of traditional laboratory screening into a few hours of computational work. This scale fundamentally changes the unit economics and the speed of R&D in longevity science.
10 trillion evaluations. I mean, that's a number that's almost too large to grasp. What does this actually mean for the future of drug pipelines?
It removed the bottleneck of molecular screening. Historically, researchers only explored a tiny fraction of the human proteome and the vast chemical space of potential drugs.
We were searching in the dark.
We were. Drug-CLIP allows researchers to screen millions of molecules across thousands of human proteins, covering around half of the estimated human druggable proteome. and it yields millions of potential active molecules. This massive, efficient output just accelerates the timeline for discovering compounds that target all those novel aging pathways we discussed earlier.
And this kind of accelerated AI is now moving from being a novel technology to just standard operating procedure, isn't it?
It is becoming infrastructural. Executive commentary in the sources suggests AI is now viewed not as some scientific moonshot, but as electricity.
It's just expected to be there.
It's expected everywhere, driving efficiency not just in the lab, but in operations. For longevity, AI is essential for mapping the messy, multifactorial biology of aging into tractable biomarkers, for stratifying cohorts, for complex clinical trials, and for drastically shortening development timelines.
And beyond discovery.
In medtech, AI is improving manufacturing yield, optimizing inventory accuracy, and performing predictive maintenance on complex medical hardware. It's everywhere.
With this explosion of AI-driven tools, safety and governance become absolutely paramount. What is the regulatory environment doing to try and keep pace?
The regulatory bodies are clearly attempting to stay aligned with the pace of innovation, which is critical for ensuring safety and maintaining public trust. The European Medicines Agency, the EMA, and the US FDA, they jointly identified 10 principles for good AI practice in medicine development.
And what do those principles emphasize?
They emphasize safety, transparency, and crucially, mitigating bias throughout the entire total product lifecycle.
Mitigating bias is a huge topic in AI. How does that manifest in the world of drug development?
Well, if the underlying molecular or patient data used to train the AI models, like DrugSLA, is skewed towards, say, a genetically homogeneous population,
Which historically it has been.
it has, then the resulting therapeutic targets or drugs may only be effective for that group. This just reinforces existing health disparities. Therefore, regulators are requiring transparency regarding training data and demanding that companies using AI adopt an aerospace engineer mindset,
Meaning what?
meaning providing audit trails and ensuring the model's decisions are auditable and fair across diverse populations.
Okay, let's transition now from the technology of discovery to the economics of Healthspan. What is the financial and social value proposition of longevity, this so-called longevity dividend?
The economic argument is powerful, but traditional GDP measures often fail to capture the true value of health improvements. The longevity dividend refers to the massive societal and financial gain that's realized when healthy life expectancy increases to match lifespan.
It's about closing that gap.
It is. The core thesis of the report is that reducing morbidity is more valuable than just extending survival in a state of frailty.
So what is the hard cash value of this health improvement? Have people tried to calculate that?
One economic analysis, which was mentioned in the sources, attempted to quantify the value of health improvements independent of just the longevity gains. They estimated the value for men in their 40s at approximately $1.2 million per capita.
$1 million per person.
And that valuation comes from accounting for reduced long-term healthcare expenditure, increased productivity, and the intrinsic value of improved well-being and function.
That certainly puts the financial incentive into clear perspective. Given that, what is the current societal cost of failing to achieve this health span goal?
The societal cost, particularly in places like the United States, is just staggering. The research team highlighted that Americans currently spend an average of 10 to 12 years of their lives in poor health. That is the morbidity window we have to close.
A decade of decline.
A decade. So functional health restoration, all the things we discussed in segment 2, is therefore an overwhelming economic priority. Because closing that health span gap reduces strain on healthcare systems, it increases tax revenue from productive citizens, and it frees up capital that's currently trapped in chronic disease management.
And how does a longer health span change the landscape of work and retirement?
It fundamentally reshapes the socioeconomic context of later life. The sources note growing evidence that older workers, particularly in knowledge-based jobs, possess superior judgment, critical thinking, and mentoring skills.
Which improves the whole workplace climate.
It improves workplace climate and stability. Furthermore, mandatory or conventional retirement often triggers decline. The loss of structure, social engagement, and physical activity can accelerate mobility challenges, the onset of chronic conditions, and mental health decline.
So staying engaged is healthier.
Extending health span allows people to stay engaged and economically productive longer, which is a triple win for individual well-being, for the economy, and for the overall productivity of businesses.
Finally, let's address the crucial issue of equity, diversity, and public trust. the ethical and social infrastructure that's necessary for these breakthroughs to actually be adopted globally.
This is the long-term challenge that limits everything else. Longevity science advancement is fundamentally constrained by public skepticism, historical mistrust, and cultural resistance.
It doesn't matter how good the science is if people don't trust it.
Exactly. The Public Longevity Group recognized this, and they have launched an initiative to develop data-driven systems specifically to measure and strengthen public trust in geroscience. They recognize that, as one expert stated, public trust is the breakthrough that unlocks all other breakthroughs.
That's a powerful statement.
It is. Without public buy-in, even the best therapies will face political and financial bottlenecks.
On the scientific side of equity, the research team emphasized the need for diversity in genomic data. They specifically pointed to the Brazilian supercentenarian cohort. Why are they such a vital source of knowledge?
Historically, our genomic datasets have been overwhelmingly focused on populations of European descent. This lack of representation creates major blind spots. It obscures unique protective longevity mechanisms.
And the Brazilian cohort is different.
Very different. It consists of highly admixed populations, a combination of European, African, and indigenous ancestries. And these individuals have shown extraordinary resilience and functionality, often reaching extreme old age, despite, in many cases, having very limited access to modern specialized healthcare throughout their lives.
So their extreme longevity is more about intrinsic biological resistance and adaptability than it is about external medical intervention.
Absolutely. Their genomes may hold unique protective alleles or gene variants that grant resistance to specific diseases or stressors. Studying them underscores that intrinsic biological factors, resistance, and adaptability are absolutely central to extreme longevity.
So we need more of this.
The research community is pushing hard to expand recruitment into these ancestrally diverse groups. It's about enhancing equity in global health research while simultaneously unlocking universal aging mechanisms that might be invisible in genetically homogeneous populations. Achieving equity in research is both an ethical necessity and a scientific imperative.
If you're finding this insightful, and we certainly hope you are, please take a moment to like this video, subscribe to Tomorrow Unveiled, and consider hitting that new hype button to help us reach more viewers. And of course, be sure to turn on notifications so you never miss an update on the future. If you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could leave us a rating and review for Tomorrow Unveiled. It truly helps us reach new listeners. We love hearing your thoughts. Leave a comment and let us know what you think about the Immortality Report and the possibility of functional rejuvenation. We've completed our analysis of the Immortality Report. Let's quickly recap the three major tectonic shifts we've seen in longevity science, corroborated across all the source material. So first, we are armed with actionable, precise tools like the IC clock and Dundin pace that move us way beyond simple chronological age. We have precise metrics of functional capacity, telling us not just how old we are, but how fast we are aging at the molecular level.
Second, the science of cellular and metabolic rejuvenation is now moving decisively toward restoration. We saw evidence of functional repair in the immune system via that HSC metabolic reboot, in the joints through precise cartilage cell phenotype shifting, and in the brain via targeted neuroprotection using GM-CSF.
These are not just incremental improvements, they're functional resets.
They are. And third, AI is accelerating the pace of discovery exponentially. Platforms like drug CLIP are converting years of traditional R&D into hours of computational vector matching. It's setting the stage for a new, rapid infrastructure that will deliver personalized therapies at scale.
Looking ahead then, what's the future of geroscience? As suggested by the breadth of this research, it seems to lie in sophisticated combinatorial therapies.
Absolutely. Aging is fundamentally multidimensional. It's not a single defect. Different organs age differently through distinct mechanisms.
So the idea of a single anti-aging pill is dead. The future is customized cocktails that address the unique aging signature of an individual.
That's the idea. For example, pairing a Senolytic compound to clear out existing debris with an epigenetic reprogramming agent to then actively rebuild and restore tissue function.
A 1-2 punch.
Exactly. This personalized approach, guided by those next-generation biomarkers we discussed in the first segment, is essential. We can measure your specific pace of aging, identify which organ is failing, and then apply a targeted, multimodal intervention to restore capacity, thereby bridging that gap between lifespan and health span.
That is the incredibly exciting future we are actively moving toward. Before we let you go, final thought for you to chew on.
The research on stress and aging highlights something pretty profound about the plasticity of our biology. We know that severe acute stress, like a major surgery, a bad infection, or trauma, can rapidly accelerate biological aging. It can visibly push back those epigenetic clocks. But crucially, robust recovery from that stress can often reverse the clock back to baseline. So if our cellular clocks are fundamentally plastic and can be reversed after acute stress, what does that imply for the long-term incremental reversal potential of chronic aging? Functional longevity may ultimately be about optimizing our body's own incredible built-in capacity for continuous lifelong repair rather than introducing something completely foreign.
A deeply provocative thought indeed. Thank you for joining us on Tomorrow Unveiled. We'll see you in the next deep dive.